We don't have too many more slides left on this lecture. All right, so let's talk about active transport. All right, so we have active and passive transport. Passive does not require energy. Active does. All right, so because we are requiring energy, it's because we are moving a solute across the membrane against its concentration gradient. All right, if we're going from low concentration to high concentration. All right, so that's unfavorable. All right, if we just had simple diffusion or even facilitated diffusion, we always want to go from high to low. We're going against the grain here. All right, so we need a source of energy. All right, so we have two sources of transport that can occur with active transport. One is primary active transport. All right, we're going to use a pump that directly uses ATP as the energy to transport the solute. Now the other one is secondary active transport, and we're not going to directly use ATP, all right, but we're using a gradient to drive movement, and that gradient was set up by the use of ATP. So even though it doesn't directly use energy, all right, it's relying on energy to set up the gradient. All right, so let's take a look at this. So let's first talk about primary active transport. All right, we have our membrane protein here. We are transporting these hydrogen atoms. All right, we're going against a gradient from low to high. So we need the input of energy, and energy comes in the form of ATP. All right, so ATP is generating energy. It's changing the shape of the transport membrane and releasing that hydrogen to the opposite side of the membrane. All right, so we're setting up this gradient where we have a large amount of hydrogen atoms outside of the membrane. All right, so if we look at secondary transport is using that gradient that primary active transport set up. Right? It's using that gradient and the tendency for molecules to want to go from high to low in order to transport a secondary molecule. All right, so let's look at our protein here. We are transporting a hydrogen atom, but we're also going to be taking in a sucrose atom. All right, now where the sucrose is going from low to high. All right, so because we're using this gradient that was set up and both of them are going to bind, that allows it to change the shape, release the sucrose against its gradient. Right? So we're not directly using ATP, but we're using the gradient that was set up by using ATP. Right? Does that make sense for everybody? All right. So a common one that we often see in molecules is this sodium-potassium ATPase. All right, so we're actively transporting sodium and potassium against their gradients using the energy from ATP hydrolysis. All right, so again, we're using ATP as our form of energy. Now in this case, we're actually setting up an electrochemical gradient. All right, so remember cells inside versus outside the cells, they have these gradients that they need to maintain. This is one of the ways that they can do that. And the way that this is done here is we are transporting these positive ions, right, these cations here, but we're not transporting them in equal amounts. All right, so 3 sodium is exiting the cell for every 2 potassium that's entering in. All right, so we have a difference in a charge. All right, so we have an antiporter, we're moving ions in opposite directions, and it's also an electrogenic pump because we are exporting 1 net positive charge out, because we're transporting 1 extra positive sodium out for every 2 that comes in. Right? So this is one way we can set up those electrochemical gradients. All right, now I threw this image in here as just kind of a review. All right, so we have passive transport requiring no energy. So that's diffusion, whether facilitated or simple. It's also osmosis. Osmosis does not require energy. Versus active transport, which requires energy, right? Whether it's in the form of ATP or a gradient that was set up by using ATP as its energy. All right, last couple of slides here. So besides just moving things inside and outside of a cell, these plasma membranes can be joined together so we can move molecules from one cell directly into another cell. Right? It doesn't have to go through any extracellular space to get to another cell. All right, so we have these intracellular channels. Right? They allow for the direct movement of adjunct cells. So in animal cells, these are called gap junctions. And we can see down here, we have one phospholipid bilayer bumping up against another phospholipid bilayer. And we have these gap junctions that allow for the direct movement of solutes from one side or from one cell into another cell. Right, now the same can happen in plant cells. They're connected by something called a plasmodesmata. And they can do the same thing. They can allow solutes to move from one cell to another cell. In this example here, we can actually see that the endoplasmic reticulum can actually be shared between two cells within that plasmodesmata. All right, so the membrane not only functions to move stuff outside of the cell, but we can move things between cells. I know I'm talking a little fast here, so I'm going to pause because I see a lot of people writing. All right, the last two concepts I'm going to talk about is exocytosis and endocytosis. All right, we briefly touched upon this, but this is used to transport large molecules, such as proteins or polysaccharides. All right, they're large molecules. They can be made up of hundreds of amino acids or hundreds of sugar monomers. Right, so we can transport it into the cell, or outside the cell via exocytosis. So material inside the cell is packaged into a vesicle. Remember we talked about vesicles when we talked about transport between the organelles like the Golgi apparatus. And it can be excreted into the extracellular medium outside of the plasma membrane. Exactly the opposite for endocytosis. This is where the plasma membrane invaginates or folds inwards and forms a vesicle that will bring substances into the cell. And there's basically three types of endocytosis that I'll just briefly explain here. One is receptor-mediated. Right? So we have receptors on the outside of our cell, and they are basically looking for very certain cargo. All right? Very specific cargo that it wants to bring into the cell. So these receptors looking for specific cargo and bringing it in. Pinocytosis. This is where we are going to basically internalize the extracellular fluid to sample it. All right, sample it to see what's around the cell. All right, this often happens around like digestive cells. So maybe think about in the intestinal area. And then phagocytosis. So phagocytosis are found in phagocyte cells, such as macrophages, and they're going to take up particles such as bacteria, right, and basically destroy that bacteria with lysosomes. Right, so it's kind of a defense system in our bodies. All right, so I'm just going to briefly show you, yeah.
What do you mean by like the extracellular medium? What area is that?
Yeah, so I mean, cells aren't directly touching each other. There's space in between, and that space has a fluid. And so depending on what's in that fluid, the cell kind of wants to find out. So it's going to take in some of that fluid and just sample what's there. Right? We're not going to get too much specifics, but I can tell it maybe it needs to synthesize more proteins to digest something quicker because it knows there's more of it outside of the cell. Right? There's a lot of different scenarios that could go on there. That's just one example. That make sense? All right. So exocytosis, we're bringing something to the outside. It typically starts in the Golgi apparatus. We're packaging it up with the help of these protein coats that's going to direct it to where it needs to go, to this plasma membrane. It's going to fuse with the plasma membrane. That plasma membrane is going to expand out and it's going to release its cargo. I'm just going to briefly go over these. This is a form of endocytosis, and this is the receptor-mediated form of endocytosis. So we see we have these receptors here that are specific for a certain kind of cargo. Once they receive the cargo, the plasma membrane will fold inward and create a vesicle with that cargo on the inside and transport it to wherever it needs to be. A lot of times it fuses with a lysosome, which can either break down the cargo into further smaller bits or just help break down that vesicle and release it to the intracellular fluid. All right, and again, with the help of a protein to help direct it where it needs to go. All right, so that was the end of the lecture that we started on Wednesday. Any questions about that lecture before we switch gears? All right, so I already went over that. All right, so we're going to start talking about enzymes. All right, so I know lab was a little ahead of lecture in that most of you have had exposure to enzymes now, right? But now we're actually going to discuss it in lecture. All right, basically two key concepts here: energy, enzymes. All right, so we need to talk about energy when we're talking about chemical reactions. So energy is the ability to promote work, or promote change, or do work. All right, and it basically comes in two forms: kinetic energy and potential energy. So kinetic energy is associated with a movement of some sort. And on the image down below, this is like the movement of the bat as you're swinging to hit a baseball. We also have potential energy. All right, that's either due to the structure or the location of, could be molecular bonds. It could be the location of the arrow within the bow here. All right, in this snapshot in time, she's just holding it still. Right, no movement is happening, but it has the potential to have a lot of energy once she lets go of that bow. Now, a big one that we're going to be talking about is chemical energy. So this is the energy that is stored in molecular bonds, particularly covalent bonds. All right, it's a form of potential energy. We're going to dive into that just a little bit further in a bit here. But there's a lot of different forms of energy in biology. Light, heat, mechanical, chemical, electrical. All right, there's a lot of energy out there. And this table is in your textbook. Just kind of threw it up there to give you an idea. So light can be energy in the form of electromagnetic radiation. Heat can be energy in the form of its transfer from one object to another. We have mechanical energy possessed by an object due to its motion relative to other objects. Chemical potential, which we're going to go over here. And electrical or ion gradient, like we just saw in active transport. Right? Sets up a gradient and allows us to perform some action. All right, this is in your books. I also threw it up here. I'm not going to go over all of this word for word. So we're going to move on to the nature of chemical energy. All right, so this is chemical potential energy. This is what's most common in cells. And this is where electrons are the most important source. All right, so electrons are important due to their configuration and their position. So if we think about back to the first lecture we had, and we talked about electron shells, right? And that the electrons in the outermost electron shell are valence electrons, and they are able to interact with other atoms to form bonds. All right, so based on where the electron is within a molecule, those on the furthest electron shell, they have the ability to interact, right? They have the ability to have some chemical potential energy, as well as their configuration. So maybe this electron is currently in a bond. Maybe it's not in a bond yet, right? So it's all kind of relating back to these outermost electrons. All right, so everybody at some point has probably learned about the laws of thermodynamics, most prominent in like physics and chemistry, but it also applies when we talk about energy and biology. All right, so first law of thermodynamics, it's one we hear often: energy cannot be created or destroyed, right? It can only be transformed from one type into another. All right, but as it's transformed into another type, this is where the second law of thermodynamics comes into play. All right, as we transform it from one form to another, it increases its entropy. All right, so entropy is a degree of disorder. All right, so if we look at our image here, we can see as we increase our entropy, we're getting more disordered. Right? And as that entropy increases, as that disorder increases, there's less energy available for organisms to promote change or do work. All right, so if we look at, say, a water molecule in a river, all right, and these are just snapshots in time. So right now we have potential energy of this water molecule, right? It's on the top, it's not doing anything, it's just sitting there. Or a snapshot in time, it's not actually flowing. All right, now we have the same molecule that's currently falling down, right? It's exhibiting movement, it has kinetic energy. All right, and finally, it gets to a point where after the kinetic energy, that kinetic energy has been transformed into another type of energy. It might be mechanical, right? That water molecule hit upon the rocks. It could be heat that was generated due to that. It could be sound, right? The sound of a waterfall. All right. It's created another form of potential energy, but at a much lower or much higher entropy and higher disorder, right? So it's a lower potential energy. All right. So energy is not created or destroyed. It's just going to change its form and change its entropy. All right. So I want to think about entropy here. All right. And as a group, answer these two questions. All right. So we have this table salt crystal at the bottom of a beaker of water. I should say of, not or. And water after the table salt has dissociated into its ions. Right. Which one has more entropy? So talk amongst yourselves for a minute here, and we'll come back together.
Okay, crystals.
All right, does anybody need more time to answer this? Yes? All right. All right, so I kind of hear the topics of conversation changing, so that cues me that it's time to come back together. All right, so which one has more entropy, the salt crystal or the crystal after it's been dissolved? I hear murmurs. After it's been dissolved, all right? Think about the organization, all right? So we have this nice compact crystal, right? We have these bonds holding it together, these ionic bonds, and water is coming in, breaking it apart, and these ions are now spread out throughout the entire beaker, all right? I think that's an easy way to visualize entropy, all right? An increase in entropy, increase in disorder. Does that make sense? All right. So we're going to move on here. So not all energy is usable energy. All right, so when we think about energy, we think about this generalized equation here, where the total energy is equals the usable energy plus the unusable energy. All right, so we know these energy transformations involve an increase in entropy or an increase in disorder. And this disorder can't be harnessed to do work. All right, so it's unusable energy. So when we were talking about the energy that we're going to use in a reaction, we're often talking about the free energy. often called the Gibbs free energy after the scientist that did the work on this. All right, but that's the amount of energy available to do work. That's the usable energy. All right, let's look at the free energy in two different types of reactions. All right, so we have an exothermic reaction, and this is where we are releasing heat energy. All right, so if we look at our Gibbs free energy here, delta in science means the change in, all right? So the book goes into a little bit more on the equation and how you actually get the Gibbs free energy, all right? But we're just going to look at the equation that's already been calculated, all right? So if that Gibbs free energy is calculated to be less than 0, this means our reaction is going to happen spontaneously, right? It's going to happen spontaneously, so we call it an exergonic reaction, all right? So exergonic reactions are common in these endothermic reactions, and that the products have less potential energy than the reactants, right? Because we're increasing entropy. Now this is different from the endothermic reaction. This is where we're actually taking up heat during the reaction, right? So the change in Gibbs free energy is going to be greater than 0, right? So this is not going to be a spontaneous reaction, and so we say it's an endergonic reaction. All right, so the products actually have a higher potential energy than the reactants. Now, a common analogy to use is think of your dorm rooms. All right, so after what has it been? 3 weeks, 4 weeks of living in a dorm room. Dorm rooms are probably a little messier than when you first moved in. All right, that's just kind of happening spontaneously, right? You don't notice it until one day you walk in and you're like, oh, I should probably clean up. All right, but the act of cleaning up, that takes energy to do. All right, so you can think about this as a spontaneous reaction. This is exergonic. And think about this as an endergonic reaction. It takes energy to clean the dorm room. All right, so there's a lot of factors that can influence our reaction rates. So for most reactions to proceed, we have to break chemical bonds. All right, these are typically covalent bonds. And once we break those bonds, other bonds have to form in order to create a different product. All right, so in order for a reaction to occur, these substances have to collide in a very specific orientation to each other that brings their electrons closer together. All right, so they're just randomly colliding, and some of those random collisions will bring them in an orientation that's favorable for the reaction to occur. Now this happens often when the temperature is very high, right? So when we have high temperatures, the molecules are moving around faster. There's a greater increase of a chance of collision in the right way. All right, this also happens when the concentrations of the reactants are high, right? We have more reactants, so we have more chances of those collisions happening in just the right orientation. All right, so let's look at the hydrolysis of ATP. All right, we have said that ATP has the ability to provide energy, say for active transport. All right, so ATP, right, we have our ATP molecule here, has a Gibbs free energy equal to -7.3, and the units on that are kilocalories per mole. All right, we're not going to get into how we got the units here or the equation itself, but if you're interested, they explain it out in the book for you. All right, but because it's negative, all right, we know that this reaction is going to happen spontaneously. It's going to be an exergonic reaction. It's going to favor the formation of products. All right, and the energy liberated is used to drive a lot of cellular processes, like active transport. All right, so that's an example of an exergonic reaction, but we can actually couple an exergonic reaction with an endergonic reaction to try to change that Gibbs free energy to be spontaneous. All right, so an endergonic reaction is not spontaneous, all right, but if we couple it with an exergonic reaction, all right, depending on what it's coupled with, we can change that Gibbs free energy to be negative, and that reaction will then occur spontaneously. All right, let's look at an example of this, because I know it doesn't quite make sense yet. All right, so we're going to look at the formation of glucose 6-phosphate. All right, so glucose 6-phosphate, That can be formed if we take a glucose molecule and a phosphate molecule and put them together, right, in the form of a dehydration reaction, right? So we have water as a byproduct, and we're forming this glucose 6-phosphate. Now the Gibbs free energy is positive, a positive 3.3. So this is not going to happen spontaneously. Now that ATP hydrolysis we just looked at, right, we know that has a Gibbs free energy of negative 7.3. Now ATP contains a phosphate group. All right, so we can combine these reactions together. All right, so we can use ATP to couple the reaction and make it a spontaneous reaction. All right, so now we're taking glucose plus ATP. Remember, it contains that phosphate group. And we're creating glucose 6-phosphate plus the byproduct of ADP. Right, and the overall Gibbs free energy is negative 4. Right, we've made it into an exergonic reaction by coupling them together. All right, so ATP, very powerful molecule. Now, these reactions, these spontaneous reactions, it makes it seem like they happen quick, all right? They're spontaneous, they happen at a moment's notice, all right? But they're not, all right? They're not necessarily a fast reaction. I think your book gives the example of gasoline and, I forget what it reacted with. But your book gives a really great example that you can have gasoline sit in a container for a while and it's not going to react with its product. But if you come back maybe after 1 million years, maybe then that gasoline has been converted into the product. All right, so it takes a long time. So enzymes play a critical role in speeding up these reactions. All right, so an enzyme is a catalyst, which is an agent that speeds up the rate of a chemical reaction without being consumed. All right, so it can be recycled and it can be reused. All right, so enzymes are a protein catalyst in living cells, and they increase the rate of chemical reactions. All right, so how an enzyme actually works, all right, is it decreases the activation energy in a reaction. So in a reaction, we have this initial input of energy, all right? This initial input of energy allows the molecules to get close enough to cause bond rearrangement. All right, so think of two molecules or two atoms or whatever you want to think about here. They have these electrons around them, and those electrons are negatively charged. All right, so negative charges, they want to repel each other. So we need that initial energy to get them close enough where they can start reacting. All right, we don't want them to repulse each other, so we have to hit them with some energy to get them close enough. Once they're close enough, they go into this transition state. All right, this is where the bonds are essentially strained out to its maximum and weakens the bonds to the point where they can break and form new bonds in order to create the product. All right, now there's many ways we can overcome this activation energy. One of them is heat. You heat up a reaction, right? That's going to help get us that initial input of energy for that activation energy. But we can also use enzymes, right? Enzymes are going to lower this energy needed. All right, so let's look at how an enzyme does that. So if we look at this reaction, all right, we have our Gibbs free energy here. We have the process along the bottom axis here. We have these reactants. This is the activation energy, and then the products are created. Now, without an enzyme, there's a high amount of activation energy that's needed. But if we use an enzyme, that activation energy lowers, right? And it's going to make that reaction happen quicker. All right, so this enzyme lowers the activation energy by straining the bonds in the reactants, making it a little easier to achieve that transition state, but also by positioning them closer together. All right, they're positioned instead of relying on randomly colliding with each other in order to get that correct orientation. They're now specifically positioned in an enzyme in the correct position. All right, so that's going to facilitate bonding to perform get those new products. All right, so we're changing the local environment of the reactions. All right, and this can happen through very temporary bonding between the enzymes and the reactants within what's called the active site here. All right, so active site, location in the enzyme where this reaction is taking place. We can see here, this is the active site where the substrate is going to bind, right, and it's going to form the enzyme substrate complex. A little bit of terminology here. And if you've had me in lab, you've probably already gone over this terminology. All right, so taking a look at the full reaction here, all right, we have our substrates binding into our hexokinase, all right, and they're oriented in a way that makes it easier to lower the activation energy and create new bonds and release our products. And again, we're looking at this glucose 6-phosphate reaction using ATP and glucose. All right, now one of the things about enzymes that is very important is that structure determines its function. And enzymes are highly, highly specific to what substrates it will bind because of its structure. All right, and it's specifically the structure of the enzyme's active site. All right, so because this active site is so specific to be able to bind a very specific substrate, if another molecule comes in, it's not going to be able to bind to it. And this allows for a lot of control within the cell. All right, so instead of an enzyme reacting with everything around it, it only has the ability to act with that specific substrate that it is designed for. All right, so this is what's called an induced fit. All right, so it's very specific for the substrates. The substrates go into the enzyme and it performs as induced fit because they're so specific for that substrate that reorients them together in a way that's going to allow that reaction to occur. All right, specificity is important here. Just a quick side note, if you ever see the suffix ase, all right, that's typically denotes an enzyme. All right, so hexokinase. All right, more terminology with enzymes here. And we're going to learn a little bit more about reactions in general with enzymes after the exam. All right, but enzymes have an affinity for their substrates. Sometimes that affinity is high, all right, so it has this high affinity to bind to a substrate even when the substrate is in low concentrations. Or it can be very weak, all right, so the enzyme can only bind to a substrate if there's enough of it because it has a low affinity. Now this reaction the reaction velocity is going to increase until it reaches a saturation point, a point where nearly all the active sites are going to be occupied by a substance. All right, so that's what we call, in shorthand, the Vmax, right? The maximum velocity. Velocity of the reaction near the maximal rate. So we have a beaker with enzyme and some substrate in it. At this point, the enzyme, all those enzymes, those active sites are filled. All right, so taking a look at that here, all right, we have a diagram of our velocity on the vertical axis, substrate on the y-axis here, or excuse me, on the x-axis, and we're going to look at the Vmax. All right, so the Vmax in this case, we're looking at the substrate concentration of different test tubes that have different concentration to them. All right, so this has a low concentration, this has a very high concentration. And we're all trying to get to that maximum rate, that Vmax, the max amount of product that can be created because all the active sites are filled. All right, now at this halfway point, we often say it's the Km, all right, where the velocity is half the maximum value. All right, I'm going to gloss over that because it is kind of important, or it is important, but it's kind of boring to learn all these different terminology. There's a lot of jargon going on. So let's look at it in terms of inhibitors. All right, so inhibitors is a molecule that's actually going to bind to that active site, and it's going to inhibit the ability of the substrate to bind. All right, so it's a different molecule, different than the substrate, binding to that active site, and now it's decreasing the halfway point of where you need to be to get to that maximum velocity. So essentially more substrate is going to be needed in the presence of a competitive inhibitor. Now we also have non-competitive inhibitors, right? They're not going to bind to the active site. They're going to bind to another site on the enzyme, the allosteric site. Right? It's going to lower that saturation rate, the Vmax, right, without affecting the halfway point. And where am I on time? All right, we're just going to quickly go through here, and you can see that they're fighting for the active site here, and they're not fighting for the active site here. So the Vmax will stay the same with a competitive one, different with a non-competitive one. Different than an irreversible inhibitor. This is where it's covalently binding to an active site, and that enzyme is done for. It's non-reversible. And the last thing I wanted to mention, I know I'm speeding through this here, is that enzymes are going to function within a very narrow range. Right? So depending on the enzyme, it's going to depend on if it functions on a lower temperature, higher temperature. Lower pH, higher pH. All right, prep for Monday. I wanted to say a quick thing about Monday. Assignment 1 is due at midnight. So don't forget about assignment 1. That is 8 points. And the exam is on Wednesday. Now on Monday, we do not have a structured class. All right, Monday is review. So if you come to class, I will have an activity for you to do if you wish. All right, otherwise come to class, ask questions. This is your review session. I will be here to answer any question that you could possibly have. I will also hand back your quiz on Monday. So if you don't want to come Monday, you're not going to get your quiz back. You can email me if you really want that before the exam. Office hours on Tuesdays, let me know.